Okay, and so our Hangout is on air now. So we are together here today to discuss the last book in our Summer of Symbiosis book club series. And we're going to be talking about Follow Your Gut, which was written by Rob Knight with Brendan Buhler. And as I understand it, this is based on Rob Knight's TED Talk that he gave. And they've kind of expanded it into a very accessible 96-page quick spin through the human microbiome and what we know about it. So we'll be discussing this book today, and we've got a good group of people here to have a lively discussion about it. So I'll introduce myself, and then I'll have everybody introduce themselves. So my name is Laura Williams. I'm a new assistant professor at Providence College. And I was interested in doing this book series. We did 3 books over the summer on symbiosis, and I'm primarily doing this out of kind of selfish reasons, because I think symbiosis is fascinating, but I'm also teaching a course in the fall, starting in 2 weeks, on symbiosis. So this was a good way to make sure that I was doing some prep over the summer so I could be ready for my fall courses. And I know next to me is Mark, and I know he's also teaching something on symbiosis soon. So Mark, do you want to introduce yourself?
Hello, I'm Mark Martin, and I'm an old associate professor of biology as opposed to a young assistant professor of biology at the University of Puget Sound in Tacoma, Washington. And as Laura said, I'm going to be teaching a freshman writing seminar about symbioses and parasitism, and I have indeed assigned this book. So I'm really looking forward to it.
Excellent. Karen, do you want to? You're right next to Mark. Do you want to see if you can get your computer going so you could briefly introduce yourself?
Sure. I hope I don't lag out. But hi, I'm Karen. I'm a PhD student at Simon Fraser University, and I study the virulence factors or proteins that cause disease in a bacteria called Francisella tularensis, which is a potential bioterrorism agent. And my interest in this book is generally I like to read really widely about microbes because I think they're really cool. So I just really enjoy reading all these books, and actually because they collect so much information and research together, it's such a good way for me to be like, oh, okay, I really want to go back and read that paper, and so I'll just end up reading like a bunch of interesting papers just because they've got me interested in it. And so that's why I like reading these books.
Yeah, that's great. That's another great outcome of a book club like this. So we're glad that you're here. We missed you last time, so we're glad you're able to join us for the last one. It looks like Emily and Kat are kind of negotiating to get both of them on the screen. So are you guys set? Do you want to introduce yourselves?
Introduce yourself?
Yeah. Hi, I'm Emily. I am starting a postdoc at the University of Alaska Anchorage with Kat, and we'll be looking at microbial community composition in lab-reared and wild 3-spine stickleback from Alaska.
I'm Kat, and I just started at the University of Alaska Anchorage as an assistant professor. Yay!
Congratulations! Okay, and last but not least, we have joining us, I think, 2 members. I can see most of Daniel and then maybe a shoulder of someone else. Aha, there we go. So I think we have 2 members of the Knight Lab. So do you guys want to introduce yourselves?
My name is Daniel McDonald, and I'm the former project manager for the American Gut and a postdoc in the Knight Lab.
And I'm Embria. I've been a postdoc in the Knight Lab for the past 1.5 years, but I'm transitioning over to be the new project manager for the American Gut project.
Cool.
Oh, excellent. Well, that's great. So thank you very much for joining us. We're really excited to have you here, especially with your perspective on the human gut, the project. So great. This is a really good group, and we're looking forward to talking about the books. So I thought what I would start with is there is a lot, as Karen said, there is a lot in here. It covers a lot of kind of what the current, at this point, is probably even a little bit, a teeny bit out of date given how fast the field is moving. But as it stands, this is a great read for people to kind of get up to date on what do we know and what are we trying to know about the human microbiome. So in the interest of kind of our symbiosis theme, I thought maybe I would ask people about their perspective on how do we view the human microbiome in the context of a symbiosis. Are there certain things that symbiosis, certain concepts that symbiosis research can help us bring to bear on understanding the human microbiome?
Well, I think it's really interesting because Seth Bordenstein apparently today has a paper coming out in PLOS about the idea of looking at us as aggregates of ourselves and the microbes associated with us. And that's one of the things that I think folks forget. They view microbes as being angels or devils, but they're integrally part of us. And I really appreciate the depth and breadth of that, you know, within this particular book. I've not only seen the TED Talk, but I've heard Rob Knight speak. And just it's dizzying the amount of stuff. And that's what's so impressive about this book, by the way, is that it does a nice snapshot for people who don't have time to go through this. And we can talk about what's cool about the book later, but really driving home the point that we are more than just a human being. We're a meta-organism. Seth doesn't like that, but we're certainly an aggregate organism. And so that's why I was really excited to see this put in such a way that's amenable for freshmen, by the way, as well as for pretty much everybody.
Mm-hmm. Mm-hmm. Yeah, I agree. That's a really good point. Did anybody else have any kind of general thoughts about the book as we're getting started?
Well, I was thinking of it in the context of the first Symbiosis book that we read. I missed the second one, but because I was in transit. But the first one that we met was all about how organelles became part of these larger cells and these more complicated cells. And when you think of the gut microbiota in that context, it's slightly different because you can actually take out individual. If you're thinking of it as, if you're thinking of the microbiota as organelles within the larger cell, you can actually, it's different because you can actually take out, you know, huge swaths of different types of microbes by hitting them with an antibiotic or changing your diet or, you know, any of the 633 things that will change your microbiome. So, yeah, so, and I think it's interesting that we're evolving, because I think about evolution all the time, so I think it's interesting that our relationship with the microbes and the hosts are evolving over time, so it's not like, it's kind of like in a, when you think about the DNA in the mitochondria and how it's become smaller and smaller, and you start looking at the DNA in these different bacterial species, and you're seeing how one microbe will lose part of its DNA because it now gets this nutrient source from another microbe, and so it's evolving to have these smaller genomes, just like you would see in mitochondria and other things like that. So I like that idea of seeing it as another organelle within this larger organism that's us.
Yeah, I think that's a really good point. And that first book that we read, for anyone who missed that, was 1 + 1 = 1 by John Archibald. And that was a really great, it covered primarily the endosymbiosis and understanding the origin of mitochondria and chloroplasts. So those are kind of like, in a way, you could maybe think of them as kind of like two extremes if we're thinking about symbiosis between eukaryotes and bacteria.
Mm-hmm. I think the evolution angle, though, is really great to highlight, though, because the gastrointestinal tract of vertebrates really evolved around the context of other organisms. And those organisms, in turn, have evolved around the context of the gastrointestinal tract. And there's quite a few very intimate relationships that have been formed throughout the years in a large number of different multicellular organisms as a result. And we're just now beginning to tease apart what a lot of those relationships are, and what those organisms that have become specialized in these environments, what they're capable of doing, as well as what they normally help to provide to augment our own genome.
But you know, it's very interesting because that's exactly right. But we're talking about an entire ecology, because we're kind of walking ecologies on this sense, and no one says, okay, what's the right predator that controls all these things on the savanna? And that's kind of where we're at with the microbiota, is that there's so many different things going on. And that's what's fascinating to me, are the examples where we can tease out one member and figure out what those are doing. Now, maybe that's reductionist of me, but there's some wonderful examples, again, in the book of exactly that. And that's going to be, I think, wonderful to begin to tease out those interactions. I mean, it is ecology.
Oh, absolutely, absolutely. And a lot of the methods that are applied for the analysis of microbiome data sets are derived from ecological principles.
Right.
Yeah. Now, Margaret McFall and I actually wondered about adaptive immunity versus innate immunity. and she wondered if vertebrates developed kind of this much more active adaptive immunity because of interactions with microbes. But you see, I tend to think of everybody as being, every living thing as being awash in microbes anyway. So, you know, that I don't really have a judgment on, but it's certainly true that we need to completely interact with all the living things around us. What's the John Donne thing, no man is an island? Well, you know, no organism is by themselves.
Right.
Yeah, that's true. So that's, in the context of that, so the book covers a lot of different, I don't want to say, I'm not really sure how to phrase it properly, not health states, but I guess like different aspects of our health and our development that can be affected by our microbes that live in and on us. So I thought maybe I would ask kind of, and especially since there's folks from the Knight Lab here, if we started with what we think we know the most about, where are we with that in terms of understanding the microbiota and the microbiome and how it affects human health and development? What do we feel like we know the most about right now?
That's a great question. I think probably what we know the most about is going to be how the microbiome affects or is associated with obesity. There's been a lot of really great research done along that arm of things, and it's been going beyond just profiling the community that's there, but trying to figure out exactly what those different bacterial species are doing and why they might be associated with obesity. And Jeff Gordon's group has done a lot of really elegant research using the power of germ-free animals. So those are animals that just completely microbe-free. And one of the best examples that I like to explain is, you know, if he takes fecal material from a lean human twin and from an obese human twin, and he will put that in two different mice, have the same exact diet, same exercise, and the mouse who received the fecal material from the obese twin takes on the fatter phenotype, whereas the mouse who receives the microbiota from the lean twin will take on the leaner phenotype. And follow-up studies have kind of showed that the reason why that could be is because the microbes that are present in the obese species are way better at extracting energy from the food. So that's one of the most elegant examples that I love to talk about. You know, one of the things that we have a problem with sometimes is we have to be really careful when we talk about causation. And we would love to say, you know, microbial community, it's messed up and this is causing this. But really what we can say in most cases now is just that there's an association. But this is one of those studies that's a little closer than most in maybe flirting with that line of causation versus association. And I think the evidence is pretty cool. We have more work to do, of course, but in terms of what I've seen, I think that's the strongest.
Yeah, I think just more generally, we probably have a much better understanding of the microbiome in mice than anything else.
Yeah.
Microbiome and what? I'm sorry.
Mice.
Mice. Okay. Yes. We can build a better mouse, right?
But you know, personally, there's a lot of challenges extending observations in mice to humans. But one of the really cool things that you can get from it is if we can observe interactions like the being able to introduce an obese phenotype just by introducing a microbial community, that suggests there's the potential for this behavior to exist in other mammals as well too. Whether we understand what the exact constituents are is a different story, but it suggests that at least it's possible.
Yeah, and do you think, so I know that recently there was a case study published that involved a fecal transplant, and it's just one case study, and it's anecdotal, but the contention was that the woman who received the fecal transplant, after the new microbial community was established, she gained weight, and she wasn't able through diet or exercise to actually be able to lose the weight again. Do you think that, like, as we're seeing more people, like, is fecal transplants, is that a place where you could start to kind of get a sense of how this works in humans? Would you be able to kind of extract any data from people who receive fecal transplants?
I think one of the big challenges there is there's a lack of specificity. You're introducing a lot of material, and whether or not we have a very strong idea of all those organisms that are going in that introduction is probably not the case right now. So I think a lot of the work that's being done right now, also in Jeff Gordon's group, is trying to identify individual constituents and grow those up into larger communities and see how those communities work in the case of being introduced into another organism. And I think that that's an easier to understand approach, because the sheer complexity of the community that you're introducing with the fecal microbiome transplant is absolutely staggering, and trying to tease apart what those relationships are, in particular in the context of the immune system or host genetics, for an individual who is already presumably sick, because I don't think you want to do an FMT if you're healthy. I think just it adds to the difficulty of trying to understand what's going on.
And I think it's very easy to carry that too far, and there have been recent publications that tend to overstate things.
Yeah.
And what I really like about the book is that they're very, very careful about it. You know, someone could say, well, you know, you're giving generalities because it's written for the general public or for an undergraduate rather than for experts. But that's a very important role, and that's why I found it so valuable. But in addition, there are fabulous references in there. And I'm not saying that to, you know, fluff it up or anything. I do think that's important. It's possible to just give kind of the USA Today version of things, and we've all seen publications like that, or it's possible to give the supplemental information in Nature approach to things. But we do have to pitch in the middle, and that's something that we all need to do as educators. Be able to let undergraduates in particular, for me, understand what this unfolding field is all about. And believe you me, folks in the Knight Lab, I spend a lot of time talking about things that come out of your laboratory.
Hopefully good things.
Well, sure.
Yeah, I think that's a really good point. And one of the things that I really enjoyed being a part of this journal club is actually talking to everybody, particularly Mark, because Mark is, he teaches me how to be careful about what I'm saying, because I do notice that sometimes I tend to make broad, overarching statements, and I'm learning to be more careful about that now. And what I noticed is even as I'm TAing, like I can teach the students to be okay with ambiguity and the progress of science. And so it's, as I'm doing it, they're being educated on the fact that science is not always sure, and so they're more accepting of unsure statements. And I just thought that was really cool. Like, I just finished TAing and they all said that it was great, and so I was like, I never thought that even just telling people as it is, even though we're not sure about it, that they could accept that as well. And I did have another point about obesity and metabolism, and so I was wondering if, do the bacteria genetically themselves already have a specific, I would call it tendency to metabolize so that they would contribute to obesity, or would they have an actual memory of being in the previous person, and then that memory is transferred with the fecal transplant? I don't know if I'm being clear with that in terms of like biochemically, but I'm just wondering if like they're genetically disposed, or if being in that previous person makes them disposed to a particular metabolism, and that might be what makes the person obese.
Karen, are you when you say memory, are you suggesting that perhaps they have for fecal transplants, if they were in a previous, if we're talking about transferring a microbial community from one person to another, or in the case of the mouse experiments, we're taking a gut community from a subject and transplanting it into a mouse. Are you asking if there were environmental cues that might have affected like gene regulation and how pathways are being expressed? Is that kind of what you're asking?
Well, now that I think about it, it's probably both. Like, it could be both gene regulation in terms of the environment of the person, or I guess both in the case of the mouse and the human. I'm just wondering, like, is it the microbe themselves that is genetically predisposed to help causing this person or this mouse to be obese? Or is it that being in an obese person makes it so that based on their diet, they kind of have these epigenetic factors that create them to have a tendency towards obese metabolism? And then, does that make any sense?
See, Karen, I think you're saying something really important, and I am not a knight of the Knight Lab, which I've now dubbed you all. But what I will say is there's this business of evolution taking place everywhere, all the time. It would not at all surprise me to think that some of our microbiota has micro-evolved to fit us. And so then the question of what happens during transplants becomes very important. What I don't know are the number of generations that microbes see within the gut, but that might be such a broad topic I couldn't answer it. But it is absolutely true that in stationary culture, bacteria, E. coli, evolves to be better at staying in stationary culture. And then there are variants that don't do that within it. So it's that whole, you step in the river, it's not the same river, it's not the same person business from, from philosophy. So I don't know if the Knights want to comment on that, but I think that's a fabulous idea.
I think there's definitely a lot of potential, even just through mechanisms like horizontal gene transfer. So it might not be individual organisms that are necessarily evolving along on a straight path, but they might be picking up some interesting genes from other members of the community as well, too. Mark, I just wanted to briefly mention, by the way, because you dubbed us Knights at the Knight Lab. So there's a former graduate student, Dan Knights, who's now a professor at the University of Minnesota. And when he graduated, there's this amazing picture of Rob Knight knighting Dan Knights, and it just says Knight knighting Knight, or Knight knighting Knights. It's pretty good.
Did you guys do that ceremony at night?
No, we didn't.
You have to go farther. I've heard a rumor that when you get your PhD in Europe, in some places, you end up getting a top hat and a sword. Well, there you go. You need to, I got gypped, right? Myself when I got mine. But you need, obviously there needs to be some kind of Knight swag for all graduates of the Knight Lab. There you go.
Did you get gypped?
Yeah, maybe I got gypped. I don't know. I don't think I got anything.
So can I ask a quick question about that? Would you guess that E. coli, and I know that's a minority component of the gut, but E. coli from the gut would have been essentially in stationary phase, or is it constantly changing within the gut? I'm curious about that.
That's a loaded question. It's so hard to answer because we're just starting to study how microbial communities are working in our systems, and the great point that Daniel brought up is that we know a lot about how things are happening in animal models. You try to take that to a human system, there's just such a higher level of complexity. So it's really hard to say. I would think, though, given the fact that the microbes in our systems are constantly exposed to changing environments, that it's not necessarily at stationary phase. And one of the reasons I would suspect that is that we have somebody in the lab who's looking at not the gut microbiome, but the oral, which is part of the GI tract. I think a lot of people forget that. And we're noting that the changes in the oral microbiome, can happen really quickly, and it depends on whether you've eaten or whether you've brushed your teeth. We've known for years now that brushing your teeth completely changes the microbiome on your teeth, so it's not at all the same thing as being a laboratory test tube. We're a living, changing organism with a lot of complex interactions, so I wouldn't necessarily think that abundance would be stationary, but then abundance is completely different from activity, so there's a lot of different things to think about when you're asking a question like that, and I don't know that we could really answer it yet.
Well, I don't want to get off on that tangent. It's just there's this phenomenon called GASP, growth advantage in stationary phase, and what I was wondering is whether or not some of the bacteria in our gut are kind of pre-adapted for that business. That's the only reason I bring it up.
Yeah.
Well, I think that. Sorry, go ahead.
Wouldn't it be based on what type of microbe you're looking at? Because the microbes in the lumen are going to be turning over so quickly as we release the microbes from our gastrointestinal tract, and then the microbes in the biofilms or in the not the biofilms in the mucus are going to be probably more likely to be in a stationary phase, just because they can survive off the mucus, they can survive off what the other microbes in the mucus are producing, and they're not being flushed out of the system quite so frequently. So I think one of the problems that we run into as people studying microbes in the gut is that there's some, not most people are really good about this. There's some I've seen some generalizations, like we're going to treat all microbes in in the gut similarly, but they're so obviously not. And I think one of the great things about studying microbiota is that you have to think of the microbes as being in this huge community where you're going to have some people who are in stationary phase and some microbes who are constantly turning over, and some microbes that are really active, and some microbes that, I actually wonder, like, are some microbes in the gut kind of like in a spore phase, and then they wake up when we eat certain things and start replicating and then go back into a spore-like phase? Or, it's really fun to think about how these different microbes interact with each other, but then interact on their own within this larger community, if that makes sense.
No, I think that's a wonderful way to look at it. And no one's that I know of has looked at gas phenomenon biofilms, which is exactly what you're talking about.
Yeah.
But Laura, I'm sorry for going off on that.
No. I think that's a good question to ask. I think these are the kinds of things where that's where this field is going, and the kinds of questions we're trying to tease out to understand how this is operating as an ecosystem. I was just going to say, speaking of diet, we were kind of talking about what do we know the most about, and the connection between the microbiome and obesity and so forth. Therefore, diet was brought up, and I think one of the things that I think is really, it was mentioned briefly in the book, and we also covered it when I taught my non-majors class in the spring. There was a study about, and this might have been Jeff Gordon's lab, I can't remember. It's the study of the Kwashiorkor twins in Malawi, where you have, and I believe they have some monozygotic twin pairs. Does the Knight lab folks, is that true?
The lab was involved in that study, and that was Jeff Gordon's group who was leading it. I believe, if I recall correctly, they had both monozygotic and dizygotic twins that were discordant for Kwashiorkor.
Right. So what you have is a pair of twins, either monozygotic or dizygotic, and one of the twins has Kwashiorkor, which is a nutritional deficiency, and one is I don't know, healthier. I don't know how you would classify them on a spectrum of health, but one did not have Kwashiorkor. And that's really striking to me, because you can assume that kind of certain factors are more controlled for in this particular study, like host genetics. So if you have monozygotic twin pairs, you're kind of trying to control for host genetics. But then if they're twin pairs, you're hopefully also trying to control for exposure during childhood and how they were raised. And you're still getting discordant health states that are associated with differences in the gut microbiome. That was a really interesting study to me, and we covered it in my non-majors class, and there were lots of questions about, well, what's going on, and what's happening, and so I didn't know if there's been follow-up to that. I haven't had a chance to look.
I'm not sure if there has been follow-up. I'm pretty sure that the Gordon group is probably continuing studies down there, because that cohort is amazing to have. I mean, individuals that are, first getting access to individuals who are in food insecure places is not a trivial task. And then developing a cohort where you have individuals who are at risk for developing Kwashiorkor and being able to monitor them. It's a huge investment to just even get that type of project started. So I would imagine they're continuing forward, but I'm not familiar specifically.
Yeah, I haven't heard of any follow-up, but I would be surprised if they weren't working on something.
So on the same lines, though, a lot of the original obesity work that the Gordon group was doing, they had a similar cohort where they had individuals who were monozygotic or dizygotic, and they were discordant for obesity. So one individual would have a BMI above 35, and the other individual would have a BMI between like 25 and 30, which is also quite difficult to get at. But the end goal is, what can you do to try to rule out host genetics as best as you can? So you're ideally just looking at the constituents of the microbial community. And then there's a bunch of other work, some relatively recent work came out, I think, last August by Ruth Lay's group. Julia Goodrich was the first author on it, where they were studying the Twins UK cohort, which is from Tim Spector's group at the King's College in the UK. And they were able to identify, I believe it was amylase gene copy number variation in the human genome that was associated with particular members of the microbial community, and this was a trait that could be passed on from the microbial community. And there's one organism in particular, I think it was Christensenaceae, or I guess it's a family, that they're able to tease out that was associated with, the presence of it was associated with a lower BMI. So that's another, along those lines of tying it back to host genetics, that work is definitely quite a fantastic piece. And it's relevant to know about if you're interested.
Yeah, that organism was by a former undergrad student of mine, Jill Waters, now doing work with Ruth Lay.
Oh yeah, I met her last year. She's awesome. She gave a really great talk at Disney.
Her goal in life was to become a poop scientist. That is what she said from day one. He sent me photographs from the Poop Museum in Korea. It's hysterical.
Achievement unlocked.
Exactly, and she worked for Abigail Salyers for her PhD, so and that just made it 1,000 times more graphic.
Oh, that's great. I think you had said that was your student. I had forgotten about that, though. So it's always great to see when your former students move on to do really exciting work.
I'm a good talent scout.
We're going to end up scaring poor Emily away from this microbiota work. Come on, guys. Either that or she's going to be so intrigued by the aspect of poop that this will just bring her in wholeheartedly. This will be interesting to see how our discussion goes after this.
Yeah.
I love fish poop.
Yeah, and fish poop isn't so bad on the spectrum of poop.
Yeah.
So, I used to work with Vibrio harvii, bioluminescent bacterium. It colonizes the fecal pellets, and that's how they're going from fish to fish, which I found fascinating.
Yeah.
So do you want to, since you mentioned it, do you want to tell us a little bit about what you're doing up there in Alaska with microbiome? It's stickleback, right?
Yeah.
Okay.
So, we have different populations of stickleback that have evolved in really different microbial and ecological niches, right? Some are in these freshwater populations that have evolved in marine ecology with lots of different microbes, and then we have some that are in freshwater environments with a very different salinity and pH and temperature fluctuations and whatnot. And these stickleback have been evolving with these different microbial communities as a result of these different environments. And so we're interested in how the diversity of these stickleback populations have evolved different relationships between the microbial community and the immune response. And then and I've got a paper coming out that I'm currently revising, in addition to everything else, currently revising. And then Emily here has a lot of experience looking at behavioral aspects of different animal models, and she has 10 years of a lot of years of experience with stickleback, and so she's interested in looking at. Actually, why don't you talk about?
Yeah, so my background is in ecology, evolution, behavior of stickleback, which I've been working on for a while. And so this is my first real foray into the wonderful world of microbes. And what's so interesting and exciting about it, as Daniel mentioned earlier, is that a lot of the basic principles of ecology and evolution that I've been working with are very readily transferable, which is really great. And as Kat mentioned, we have at our disposal here in Alaska this wonderful natural experiment, this adaptive radiation of stickleback. So what I'm interested in, not only looking at natural diversity in wild populations, but we're also potentially interested in looking at how microbes might be influencing behavior in stickleback. So behavior has been widely studied since the days of Nico Tinbergen. So we know a lot about how stickleback behave both in the wild and in the lab. So a lot of behavioral assays have been developed. So this is an avenue that we might be interested in getting into. I know some work has been done in insects looking at assortative mating and how microbial community influences mate selection. And things like that would be interesting to look at in stickleback as well, because we know that assortative mating is very common in a lot of populations that we've looked at. So I'd be really interested to get your point of view, all of you, poll the audience, on what you've been thinking about maybe, what you've been reading about with regard to how microbes may be influencing behavior.
Well, First, Emily, by the way, good to see you again.
Yeah, good to see you too. It's been a while. We met in Chester-Krumlov about a year and a half ago, 2 years ago almost.
I think it was a year and a half, 2 years ago.
Yeah.
So Emily was at a workshop called the Workshop on Genomics that takes part every year in the Czech Republic. Have you guys looked at the Antibiotic stickleback that Bill Cresco's lab is able to produce?
That's
her.
That's me. I developed that.
Oh, awesome. Okay.
Yeah, that's the model that I'm bringing here. So we can make our fish germ-free, and then we can add back individual microbes, or you can add back complex microbial communities, and we can also make the fish, make the bacteria fluorescent, and we can follow, because stickleback juveniles are transparent, relatively transparent compared to a mouse or anything else. We can actually follow these fluorescent bacteria in a live fish. And so we can start looking at things like dynamics of microbial communities in these populations that have different genetic backgrounds. So how does the host genetic background influence how these microbial communities both form, and which microbial members are kicked out or which ones thrive in these different microbial, in these different stickleback with different genetic backgrounds.
That is fascinating, and you have large sample sizes as well too, which is a traditional program of doing notobiotic hosts.
Yes, we can get up to 120 eggs at a time. So from a single crossing of a male and a female. So that's adds a lot of statistical power to our analysis, which I'm really excited about.
Yeah, it's great.
So I'm really glad you spend some time to talk a little bit about your system, because that's your question about the microbiome and behavior. That is one of the things that I was going to ask about, because I feel like that's probably on the spectrum of how much do we know about this. How the microbiome affects behavior is probably on the we don't really know a lot yet side, I would say.
I think we know very little about even just the genetic basis of behavior. Behavior is a complex trait. So it's been really hard to tease apart, environmental versus genetic effects. So it's still a bit of a black box for sure.
Mm-hmm. Yeah, it sounds like a really fantastic system. I mean, having notobiotic stickleback and being able to track them and have a fluorescent tag, that just sounds really great. So that's very exciting.
Yeah, it's gonna be fun.
You do know that there's a comedy video where they took Justin Timberlake's Sexy Back and called it Stickleback. You did know that, right?
Yes.
Oh, yeah. Anyway, I was curious what you thought about Mazanian. I want to be able to say his name correctly, and it's funny because my sister-in-law is Armenian. Mazmanian's work, and what I like about that idea with behavior is he has a model for it having to do with inflammation, as opposed to there's an association between these different phylotypes and changes in behavior. What are your thoughts there?
I haven't read his work on behavior. I'm familiar with his work. You're talking about Sarkozy?
Sarkis?
Sarkis.
Yeah. I'm familiar with his work with PSA, and but I haven't followed his most recent work looking at behavior and how the microbes affect the behavior. I'm assuming he's doing this in his mouse model?
Yes.
Yeah. Okay. Add that to our to-read list.
Yeah.
So Knights, I know you've had some involvement with that. What are your thoughts?
Well, so Embria was involved in the autism work that was done with Elaine Shu.
Yeah, I actually was involved in that before I came on as a member of the Knight Lab. I worked on it during my PhD. It was an interesting side project. Sarkis, his group, they're great to work with. I think the work that Sarkis is doing, and now Elaine, since she stepped out on her own, is really interesting. But again, it's a great example of one of those things that is really easy to sensationalize, and you have to be very careful for a number of reasons. The first is that, again, we're working with animal models, and it's really, I think, probably anybody working with microbes in animals know that a lot of times you can't 100% recapitulate a human disorder in an animal model. And so we're looking at mouse models of autism, and we're saying that these mice are autistic, but it's really just based on an abnormal behavioral phenotype. So there's no way to know 100% whether, how do you say a mouse is autistic?
Well, No, I'm not trying to do that. I've heard the gentleman say that the bad part is he would get letters from parents and how heartbreaking that must be.
Yeah.
I think what I was trying to get at is that when you talk about behavioral changes, there's a good model in that particular one having to do with a dysbiosis that leads in turn to inflammation. Go ahead.
Physiological changes that Sarkis and his group saw upon giving the Bacteroides fragilis was pretty drastic. The leaky gut pathology was reversed to an extent, so I think there's definitely some good evidence there, but we really need to tease apart exactly what is going on. And I think, based on what I know about this study, it's really similar to what we see with probiotics. In some cases, we give a probiotic organism, the organism doesn't stick around, but in certain people you might see a change. And that's exactly what we saw in these animals. The organism that they were given didn't stick around, but the group was still able to detect some changes in the phenotype. So that's interesting and just gives more clues to the real high level of complexity that we're looking at when we study these types of things.
Yeah, I think that's there's so much there in terms of what could possibly, 5 years from now, I think we'll be at a very different place than we are even right now. 2 years from now, we'll probably be at a very different place than we are even right now. But
I was going to say, Laura, I remember when enterotypes everyone talked about them all the time.
Yep.
And I used to make jokes about it. We needed to found a company called eHarmony for enteroharmony, matching people on the basis of their microbiota. But I don't think that's such a good idea. But again, people really get carried away very, very easily on that. And I really like how cautious this book is on that subject.
Yeah, because I think it's very early stages for trying to understand what the possible effects are, and trying to tease out all the confounding factors and figuring out how. Because as Emily said, behavior is very complex. It's complex in sticklebacks. It's definitely complex in humans, and so this is the chapter on the gut bacteria and mood is a short one, and I think. I think that's okay because it's exciting and it's new, but I think it's early days.
Right.
So.
One of the interesting things I learned actually just relatively recently, so the concept of like leaky gut syndrome, those tight proteins or tight juncture proteins or whatever breaking down or becoming looser effectively and allowing more things to get into the bloodstream. What I didn't realize actually is those are the same types of proteins that are involved in the blood-brain barrier. And I'd always assumed that the blood-brain barrier was actually a static thing, but it's something that can become more permeable or change its permeability throughout your life. And so there's at least a lot of potential for things that are in your bloodstream for what actually is making it across the blood-brain barrier to change over time. And of course, possibly related to inflammation. But also related to that is in your bloodstream, I've heard estimates of somewhere between 10 to 20% of the metabolites that are in your bloodstream are derived from the microbiome. And so if you have a situation where where your blood-brain barrier can actually change over time, potentially due to some condition or some precondition, then you can potentially alter what is actually making it across to your brain, in which case there is a very direct relationship between what your microbiome might be producing and what can actually influence your brain. But it's all potential right now, and we can begin to observe some of these things in mice, but we need to understand what the basic concepts are in relation to neurological disorders and the microbiome before we get really, really, really excited. But there's at least a lot of very exciting potential, which is fun.
I know that in Oregon, Judith Eisen Lab and Karen Gilliman are working together to look at, they're using zebrafish, because the cool thing about zebrafish is you can, fluorescently label all these cells and then watch their development in the live fish. And so one of the things they're developing is using notobiotic tools to follow neuron development. And I think that's going to be a really important contribution to knowing how well the microbiota affects, or how much the microbiota affects neuron development, and then even in later stage, how are the microbiota affecting the stability of the neuron and the communication between individual cells. And I'm excited about the zebrafish model that they're developing. Yay, fish.
Yay, fish. That's the conclusion, right? Okay, so I thought one of the things that I wanted to kind of make sure we covered today was the connection between antibiotics and specifically the gut microbiome, because that's where I think most of this work has been done. I read Missing Microbes, and I think, did you read it as well?
I wrote a review of it, in fact.
That's right, okay. And I'm not sure if Karen read it also. Karen, did you read it? I can't remember.
Yeah, we read it together.
That's right, yes, sorry, I was like, wait a minute. Sorry, it's been a long
Where's Dave
year.
Baltris when you need him, right?
Yeah, yeah. Well, so it's interesting because it's clear that there's a relationship between antibiotics and the microbiome, and of course there would be, because the antibiotics are trying to kill microbes. So that makes sense. And then beyond that, I think there's a lot going on in terms of what are the long-term impacts of antibiotic treatment. And then because of that, how do we make decisions about prescribing antibiotics, when antibiotics are taken? I think I saw something recently that was an argument that for a long time the party line has been you should finish your entire antibiotic course. And then I think I saw something recently where somebody argued the reverse, where they were like, no, you shouldn't. You should take it until you're better, and then you should stop. So it seems like there's a lot to tease out in terms of the relationship between antibiotics and the microbiome. And I think this is kind of where I think we're seeing a lot of things get translated into advice being given now for what you should do. Like, should you have your kids take antibiotics for this? Well, maybe not, because we think it might have these long-term impacts. But I don't know that we know yet what the long-term impact is of antibiotic treatment on the health of the host, really.
Yeah, but I mean, if your doctor recommends that you take some antibiotics, you should probably listen to your doctor, as opposed to listening to a news article or science that is very well described as basic research. So antibiotic studies are very difficult in humans because it's very difficult to write a human subjects research protocol that gets approved in which you say, I'm going to give very hardcore antibiotics to individuals who are otherwise healthy. And so there have been a few studies that have been done, but the sample sizes have been quite small. But the basic observations from those studies is that if you give the same antibiotic to multiple people, you observe different effects on the microbial community. If you give the same antibiotic to the same person at different times, you observe different effects on the microbial community. And what it suggests is that we really, I don't think, have a great idea of what antibiotics are actually doing, or even the different classes of antibiotics, what they're actually doing to the microbiome. And it is extremely premature at this point. What Marty Blaser is getting at, though, with the Missing Microbe book, though, is really the more general concept that by having a large number of antibiotics introduced in our environment, either through for medical reasons or agricultural reasons, that there's a lot of potential to be driving more change in these communities than might be good. So that's my take with that, I guess.
No, I agree completely with that, and I mean, I believe the E.O. Wilson island biogeography is all about nuking islands with pesticides, and in some ways that's kind of what we do with antibiotics, and look at what grows back. And that is, and everything you're saying about the difficulty in working with humans is certainly true, but isn't it silly that we've done the other part of the experiment completely willy-nilly by treating people in very high numbers, using it in agriculture so much? But I don't want to turn it the other way, where there's a suggestion that you roll your children around the subways in New York, as I recently
You don't do that? People don't just do that?
There's a wonderful bit in this book, too, about Rob Knight's daughter, which I thought was fabulous, about making sure she was exposed to the correct microbes. And I do think that's true, but it takes us to that angel and devil business again, which is, I think, very dangerous. Microbes aren't all bad, nor are they all good. It's an ecology, and there are leopards, and then there are breadfruit trees, and all the things in between. It's the context between things. Mm-hmm. And that's what makes the Knight Lab have the challenges that it does, by looking at all. I mean, you want to talk about real systems biology. That's the gut. Am I right?
Yeah.
Well, I mean, you probably want to factor in, the immune system,
Yeah.
and host genetics, and all the other major components of the human body. But it's a big factor for sure.
But it's funny you bring this up, because when I talk about effects of microbes on behavior for some of my classes, I bring up that hoverfly business, where the aphids are producing a semiochemical that attracts the hoverfly and it works in the field. And I brought up the true story about the science fiction writer H.G. Wells, who was quite the ladies' man of his time. And when they asked in his biography, and I can show you the passage, it's great, they asked some of these society women what they found so attractive about him, because he wasn't a traditionally attractive person. They all said he had an interesting smell. And it makes you wonder about the role that microbes have in our behavior. But then I've had students say, well, all you have to do is find that microbe and sell it as an aphrodisiac. And I said, it's like the real estate agents say, location, 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 right? And I mean, that's the danger in all the studies of the microbiome, and why what we teach is so important. To teach it that it's not just one organism, it's not reductionist, it's integrative to everything. It is ecology. And I'm not even an ecologist, and I'm saying that.
So, and that's a good kind of point to think about all of these different issues that the book covers in terms of the relationship between the microbiome and either mood or obesity or different health states. And so, since we're coming up on 3 o'clock, and I don't want to keep people too much longer, I thought, kind of following up with what Mark said, if people had some final thoughts on where we're going from here, based on what's covered in the book. What do we think is the most important thing to keep in mind as we're moving forward with microbiome studies, either what we're focused on in the lab or how we talk about it to the public?
Well, one of the things I liked about this book is that it didn't just talk about the microbiota as this one thing that, it goes back to this, the whole theme of this book club is that it doesn't just talk about the microbiota as this one community in the gut. It talks about this this community in the concept of this much larger being that's the human, that's the host of all these different microbes, and how they're interacting with each other. And I thought that two things. One is that this book is going to be my primer for people coming into my lab, like Emily. Emily had to read the book, and then I like the fact that it's just a jumping off point, because it does place the microbiota in this larger context. And then, but it's not just within the host, it's between hosts, so how does one host influence another? If you're in a family where people are genetically related and they're eating the similar things, but you still have one person who's very thin and one person who's, considerably heavier, and how does the microbiota affect that? But how does everything else contribute to that? What they're eating, which one played on the farm as a kid versus the other kid who grew up in the city in a more sterile environment or whatever. And it's really one of the things I like about this book is it really places these microbial communities in this much larger context in a way that the public can understand it, so that they're not just overwhelmed by the science of it all.
Yeah, I think that's a great summary. Did anybody else have any thoughts about the book overall?
I find that it's important to continue to realize that this is a very young science. This is all very new to basically everybody who's doing work in the microbiome field, and there's so much that we don't know. We have some idea of what microbial communities look like in humans. Some idea, not a complete idea. Like, I can't tell you what a healthy microbiome composition is. I could tell you what some diseased ones look like, but I can't tell you what a healthy microbiome composition is. But I could give you an idea of what one might look like in North America. I don't know if that's going to be the same as individuals who are living in Africa. But really, outside of humans, I mean, we just have a very bad idea at this point of microbial life in general. So the tree of life has a very large number of phyla that are associated with either bacteria or archaea. And I think we have sequenced genomes from maybe 30 or 40 different phyla. But from uncultured little pieces of DNA, we have evidence to suggest that there's upwards of 90 different phyla. I mean, just our perspective on microbial life alone is so small at this point. And while we do have a little bit of a better idea of the types of organisms that typically reside within at least individuals within North America, it's still quite premature at this point. And there's a lot of exciting research to be done going forward.
A couple years ago, Caitlin Reese did artwork for my microbiology class, and she came up with this beautiful picture of Frank Sinatra made up of microbes, with that famous quote from Dean Martin that, they used to say, it's Frank's world, we just live in it. It is a microbial world, and we just live in it. And it's not surprising those interactions run in both directions. One of the things that I took away from this and that I think about in the future is not simply what is a normal microbiome, but what is an ecology within us. What is the gut? Well, that depends on what part of the gut. That's true everywhere. There was wonderful discussion in here of how anaerobes tend to colonize tumors, for example. We are walking ecologies all the time, and there are different microenvironments everywhere. It's the challenge to ecology. It is indeed a very young science. But I think this is a wonderful entry point, because what we suffer from in science when things get popular or scientifically sexy is people get carried away. And I really appreciate the caution with which this book was written.
Yeah.
Me too. Yeah, definitely. Karen, do you have any final thoughts before we part?
No, I'm good. My laptop's actually running out of battery, so I will probably be gone in the next few minutes.
Okay. Okay. Well, I completely agree with the final comments that Kat and Daniel and Mark made. I think this is a really nice. I'll hold my copy too. I think this is an excellent book for people who are interested in understanding what's going on with the microbiome. I keep seeing news articles about it. I just want to understand where we are. This is a really great resource for people. So I might be giving this to people for Christmas in my family. And I'll make them read it so that they know what's going on. But I think it's accessible for the public. It's accessible for students. But I'll say too that as an active microbiologist, this was really good for me to get kind of a grounding in where are we right now and where are we going, as I keep seeing things, come across my Twitter feed or come across blogs about different studies that are happening. So it's a really exciting field to follow. I'm looking forward to seeing more from the Knight Lab. I'm looking forward to seeing what Kat and Emily do in Alaska, because that sounds fantastic. So we'll be watching to see what comes out of all of that. And I want to thank everybody for joining us today. This was a really great discussion on a really interesting topic. So thanks to everybody for being here.
Thank you for coordinating, Laura.
Yeah, of course.
Thank you for this opportunity.
Yeah, thanks for having us.
Absolutely. I'm really glad you could join us. And this is our last Summer of Symbiosis book club. So we'll be, normally what we do during the school year is we have a hangout journal club that meets about once a month. So if anybody ends up watching this on YouTube somewhere, which apparently people do because we have more than like single-digit views, so good job us. We'll probably resume our journal club, so people can watch for that if they want to talk about papers. And thanks to everybody for making this a really interesting book club series that we've done. So with that, I will bid everyone adieu, and thank you very much.
Thanks.
Thanks.
See you.
See you.